From Microbe TV, this is Immune, episode number 91, recorded on February 25th, 2025. I'm Vincent Racaniello, and you're listening to the podcast about the body's defenders against disease. Joining me today from Ithaca, New York, Cindy Leifer.
Hi, welcome back. At least it's not freezing. So it's progress.
Cindy was briefly at the incubator last week.
That was super fun.
It's fun. You've been here before, though, right?
I have. One time at the opening. I did drive down and then spent the night and came back.
Yeah, I remember that.
Yep.
Also joining us from Cleveland, Ohio, Steph Langle.
Hello. Hi, everyone. Great to be here.
Who has never been here at the incubator.
No, I need to come. I'm coming to New York in early May, so I will contact you.
Okay.
Yep.
Hopefully I'll be here. Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. Sad that I'm not at the incubator today, but I have been over there. Have to come back sometime soon.
You have been here. That's right. You were here for the fundraiser as well.
I was.
And,
yep.
and TWIV 1000.
Mm-hmm.
Yeah, that was cool. We have two guests today from the University of Georgia. Where is that? What's the city?
Athens.
Athens, right. The temple. From Athens, Georgia, Klonowski, welcome to Immune.
Thank you for having me. It's great to be here.
And also from Athens, David Peterson, welcome.
Oh, thank you. Great to be here.
I visited there a couple of years ago. I visited UGA. You know, my TWIV co-host, Kathy Spindler, was there for many years.
I didn't know that.
Were you there when Kathy was there?
I don't recognize the name, I hate to say, but I know at least she was not in our department, because if she was and I don't remember, there's a problem.
Yeah, David and I are in different departments. David is in the College of Veterinary Medicine in the Department of Infectious Diseases, and then I am in the Department of Cell Biology, which is in the College of Arts and Science here. So we have different groups of people.
Do you have a biology department?
We do, and it's mostly instructional. So
maybe that's where Kathy was.
Unlike a lot of, at least universities that I was familiar with, the biological sciences are really divided here at UGA. So there's a genetics department, a microbiology department, a biochemistry department, a cell biology department. Who am I missing? Biochemistry. Did I say biochemistry? Yeah. So all the sciences are really subspecialized.
I've been there twice in the last 10 years. And the first time we did a podcast in some kind of classroom. And there was a skeleton of a dog right up front.
It's probably the vet school,
maybe David, you know about that. Maybe it was the vet school. I don't know.
Could have been the vet school.
But one of my guests worked on rabies, so it was appropriate.
Very good.
Yes, and probably one of your colleagues, David.
So my question is, how big was the dog, Vincent? Because they do have a full skeleton of a rhino.
No, it was, I would say, it was about 2 feet high.
Okay, so not the rhino.
It wasn't a rhino. But I put it right next to me, and I would pet it now and then. So one of my guests, Biao Hu was one of my guests.
Mm-hmm. Oh, yeah.
Which you probably know him. He actually invited me the first time, and then the rabies guy I don't remember. But it was a lot of fun. We had a good time.
Yeah, I had the pleasure of visiting last Monday and was able to meet with David and Kim. And it's warmer down there. It's a lovely visit. Really enjoyed my time.
We had the Society for Leukocyte Biology conference there 2 years ago, I think it was.
And ASV, yeah.
ASV was here, yes.
Oh, yeah.
Was there too, yeah.
It's a good place to go. You do have to get on a bus and go on an hour and a half from Atlanta. So it's a bit of a journey. But it's very, it's very nice.
When I first time I went, I took that bus, and for some reason the buses in front of us were carrying the local football team, the Falcons, and I mean everybody was going nuts, and the driver wanted to follow them. I'm like, can we just get there? What are you going to do if you follow them? You can't see anything, right?
Is it the Bulldogs, not the Falcons? Right?
No, the Falcons.
Yeah, that's our Atlanta team.
Oh, you're saying the pro football was in front of you. I see. I was
had like 5 buses and they had Falcons on the side. Okay. Everyone was so excited. Oh my gosh. If we could only get them as excited for science as for football. People complain that our podcasts are too long. And I say, really? You watch 3 hours of football? And it's not too long? But that's entertainment, So we have to be more entertaining.
I guess.
Speaking of entertainment, if you like these programs, which are about science, if you want real science, here's the place to come. We don't fake any science. We don't tell you lies. We don't tell you that vaccines don't work, which is ridiculous.
Yeah.
But apparently that's what you need to do to get confirmed for a high-level position, is tell lies. But here on Microbe TV, we give you great science. We need your support to keep doing it because we don't do ads. We don't do anything except get your contributions. So, and you can have a tax deduction if you live here in the U.S. So go to microbe.tv/contribute. Let's start before we hit our papers. Let's find out a little bit about you guys. Kim, give us your history.
Oh, my history. So
all the way back.
All the way back. Okay, so I was born and raised in the state of Delaware. I was always interested in science. I did not have scientists as parents. My dad worked at a factory. My mom was an administrator at a university, which ended up being very fortunate for me because her employment allowed me to go to the University of Delaware for free.
Oh, nice.
So that was wonderful. I loved being there. I was a biology major. Wasn't sure exactly what I wanted to do. Of course, as like most, prior to college, I think like most kids, there were points in my kind of thought process about science was, maybe I can be a marine biologist or maybe I can be an astronaut. So I am a certified scuba diver.
Wrong direction.
Wow. I also am an alumni of Space Camp. So I did end up dabbling in all of those things. But when I was at the University of Delaware, I also had a minor in what was called medical humanities, which was a really great minor because it went through things like the history of medicine, ethics in medicine. It was really a great minor. And we, at this time, I guess I'm going to date myself, it was just past kind of the start of the AIDS epidemic. So we were really learning about we had learned about HIV and the AIDS crisis. And I think I learned more about it through my minor in terms of like policies and things that were taking place. But then I just got really interested in the science behind it, right? And of course, this is a disease that affects the immune system. So I decided, heck, I'm going to take an immunology class. Then I went right to the immunology, not even the micro with a little dabble of immunology, right to the immunology. And I loved it. It was fascinating. So then I decided very last minute, I didn't have a ton of research experience. I had been working at AstraZeneca in the summer, really making money and doing research, that I was going to apply to graduate school and see what that was all about. So I went to Temple University in Philadelphia. It was local. I worked in a lab studying atypical VDJ rearrangements and how they contribute to the development of autoimmunity. So not anywhere close to what I'm doing now, but it was a great experience. I had a great mentor, Mark Monestier, who was wonderful. And then I decided I wanted to get away from the molecular immunology and do some more cellular immunology. So at that point, I decided for my postdoc that I was going to go to the University of Connecticut and work with Leo Lefrancois, who was really studying CD8 T-cell memory at the time. So I worked in his lab doing my postdoc. And I have to mention also at each step on the way, I really didn't know what I wanted to do. So it was like, do I want to go into industry, policy? I was like, I don't know. I'll keep going further, keep training more, just train more. And then I just decided as I was getting near the end of my postdoc, like, I think I really want to do academia. And that's kind of where it landed me and where I am today. So maybe that was too much information, but that's is the whole history.
Was that your first position at UGA and have you stayed there or have you moved around?
Yeah, no, I've been here. I've been here ever since. So it's a great place to live.
So I knew when I saw the podcast notes that I recognized your name, and I was trying to figure out why that was. And I also did a PhD on CD8 memory, and I'm pretty sure I cited a bunch of your stuff in my dissertation.
I love that. Love that.
That's awesome.
Nice. What about you, David?
So I also had parents that were not scientists. I was born in Chicago, but we moved when I was, 2.5 to sunny Southern California. So I grew up there. I was one of those nerd kids that always was into science. I had the chemistry set at home. I even had the radiation kit. They sent you a little pin, and on the head of the pin was a bit of radium. I don't think they could do that anymore.
I'm going to guess
you could make a cloud chamber and see particles as they passed through the.
Cool. Oh,
yeah,
cool.
That was exciting. But in high school, I did really well in science classes and enjoyed it, and went to University of California, Davis as a biochem major, and got to work in a lab. I don't remember how I decided to work in a lab as an undergrad, but I worked in the lab of Erwin Siegel, who literally wrote the book on enzyme kinetics. I still have it somewhere. It's about 4 inches thick. And working in his lab, I worked with some of the postdocs in his lab, and I just loved it because they kind of gave me a project and would check in on me, but I pretty much got to work out the whole purification scheme for nitrate reductase from Penicillium chrysogenum. And it was cool. I just loved it. And so when, time to graduate, I decided I wanted to go to grad school and did that in the UC system as well down at UC Irvine, which is where I started working on parasites. I worked on Trypanosoma cruzi there. The particular protein I was working on turned out to be a member of a giant multi-gene family, just a pain in the butt to work with that. And I said, okay, that's the last time I work with this. Fate has a funny way of getting back to you, though, because it turned out my postdoc at NIH in the lab of Tom Willems, I ended up working. I didn't know it until I was deep into it, but I was working on something that was part of a multi-gene family. So, and I still work on that to this day. And it's this is in malaria, Plasmodium falciparum, and that multi-gene family is involved in antigenic variation, and a lot of has to do with pathology of the disease as well. So, and still working at that. So,
we have a podcast called This Week in Parasitism.
Oh, I know.
So we should have you on that sometime to talk about your work.
That'd be cool.
Unfortunately, Dixon's no longer with us. It would have been. He would have loved to hear it.
Yeah. Yes.
But hopefully the podcast keeps going. So there was nothing like. Did you ever meet Dixon?
I think I did once.
Yeah.
Yeah, I think I did.
Steph, you wanted to know about their podcast, right?
Yes. Well, I would like for them to tell us what their podcast is. I had the honor of being on it last week. So either Kim or David, feel free to share what it is, the target audience, and maybe some anecdotes of what you've learned and if anything surprised you in the process.
David, I'm going to let you start since it was
so
or less your baby.
Yeah, the history of it was that Don Harn in our department, who is an immunologist and works on parasitic worms, had an idea for this and approached me about it. And we found another faculty member that we could convince to come on. And I have to say, it was rough starting because you listen to people on the radio or something that are really good, or in podcasts that are really good at interviewing people. And you think, okay, I think I kind of know how to do this, which is the equivalent of watching a golf pro hit a ball and saying, okay, I've never tried that, but I think I got it down now. So there were some technical issues. There were just kind of learning how to keep a conversation going and learning what are the interesting questions to ask. And then our original host, our co-host on that, Liliana Salvador, left. And that's when Kim came in a year and a half ago, I guess.
Mm-hmm. When did you start overall?
It was in, I think, fall of 21 or maybe 21, because we kind of ended up, after Dr. Salvador left, we ended up stopping for a while, and then we wanted to get it back going. And we recruited Kim because I told her that, no, it really didn't take much time to.
Got her in through that way.
No, it's really fun. And I really enjoy it because I think, I'm talking to people who I'm sure believe the same thing I do, is that it's probably important now more than ever to talk to people about science. And I think David and I were pretty together with the idea that we wanted this podcast to be accessible. We wanted it to be for scientists and maybe even non-scientists who want to learn a little bit about science. And that's been very important to me. Would you agree, David?
I agree. I want it to be something, say, that prospective grad students tune into to maybe figure out, if not, a particular professor they might want to work with, but a particular area. So, and even undergrads, if I've recommended it to undergrads in some of my classes, because I tell them, you're at a tier 1 research institution, you ought to be in a lab doing some research.
And the focus is you started, and first of all, I don't actually think we said the name.
I was going to say that. I like the name. You should say it.
People, Parasites, and Plagues. Is that accurate? Yes. Okay, good. It's a great title. It's a great name. And your original goal was mostly to stay within the UGA system to interview those on campus, but maybe also a little bit off campus as well. Is that accurate?
Yeah, we have some satellite campuses as well. So, having some of those folks on. But that was really the idea, is to kind of highlight some of the research that's been done here at UGA. And, those are easier guests to get maybe than those from other institutions. But I think we've been broadening. I mean, Stephanie, we had you on the podcast. We've had some of our seminar speakers. We want to find, we're still in those early days too, in some ways. So we want to make sure we have guests that we're going to be really fun to have on too. So we try to be probably a little bit more selective with who we have. We have everybody on, of course, that we want to, and there's lots of people who fit that description at UGA. But when it comes to having some people outside of the UGA, we try to make sure that we have somebody that we think that our listeners in particular are really going to enjoy listening to.
So it sounds like your format is pretty interview focused. Is that true? And do you have specific disciplines or topics that you try to focus on? I could imagine People, Parasites, and Plagues being super science focused, or I could imagine it being going into some of the history and some of the medical humanities that you mentioned. So which direction is it?
Well, it's interesting that you mentioned history, because one of our recent guests was someone from the history department here, Dr. Susan Mattern. And she had written a book on the Roman physician Galen, who he didn't invent the 4 humors theories of diseases, but he certainly popularized it. And it was really interesting to have her on and get a take on something that really was the guiding principle of medicine for over 1,000 years. And we've also had folks from our Grady College of Journalism, Glenn Nowak, who is expert in science communication, used to work at CDC, trying to convince people to get vaccinated. And that's really interesting. But yeah, it's interview focused, and we try to come up with questions. And actually, one of the most interesting questions is one that you guys kind of started us out with, which is, how did you come to be in science? And we've had so many just great answers. And so that's the standard we always start with now.
I think it's a great question because as you said, everybody's journey is different. And I think it's really important for trainees to hear that, because they always think there's one way to do this, one way to get to being a professor or whatever. And to hear that so many people had so many different circuitous routes of getting there, I think is really fascinating.
I think what really made us decide to always go with this question, it's one that we didn't ask this individual, but at the end of the podcast interview, she said, there's something else I want to touch on. And that's the fact that I didn't initially graduate high school.
Oh, wow.
I got a GRE a couple years later, and then I worked at a place, and then I got really excited about something, and I decided to go to grad school. And it was so inspirational, we kind of split it off as a separate mini podcast. And from then on, we've always gone to that question.
It's when I started these podcasts, it seemed the obvious question to ask people, right? I was interested myself, and then people really liked it.
Yeah. By
the way, your title, Dixon wrote a book called People, Parasites, and Plowshares.
Oh, yes. Yes.
It's a very interesting layperson's book about different parasites in each chapter. Yeah.
I'm going to have to look that up.
Mm-hmm. It's good alliteration. Well, good. Well, thanks for sharing that. I think people can check that podcast out as well as ours, and get to hear about the research going on at UGA. So we thought we'd let our guests pick 2 papers that they could introduce us to and describe some of the work being done in each of those. And I don't know who wants to go first. Maybe we'll let you 2 decide between yourselves.
Kim?
Go. You want me to go first? Sure. Thanks. So I picked up a paper that's a little bit outside of my area of expertise, but I think it's a really important one that deserves some discussion or some press, I guess I should say. And this paper came out of the Hoquist lab, and it's really speaking to one of the key tenets of immunology, which is the ability to differentiate between self and non-self, right? So that's what really essentially keeps us alive. It's how our immune system keeps us alive. The paper is really following up on this idea that had been reported by other people as well, that type 1 interferons, or there was an interferon signature that was observed in the thymus. And the thymus is really the school for T cells, right? So where T cells are trained to recognize self-MHC, but then also not recognize, self-antigens or self-peptides very strongly. And it was very weird that this kind of the signature that's normally associated with an infection was being measured in the thymus. And what they found was that the cells that were expressing the type 1 interferons were thymic epithelial cells, which are the key cell that plays an important role in educating the T cells. And what they found was that, and I'm speaking just kind of very highly about this paper, or high level, and what they found was that these type 1 interferons were not due to the presence of an infection or the microbiome. It was really a sterile inflammatory signal. And what it was doing was really two things. The first was it was activating some antigen-presenting cells in the thymus, because we know those cells also play a role in addition to the TECs to educating the T cells. But it was also believed to be playing an important role in what has been referred to since this paper was published as the inflamed self. So the idea is that when we train T cells, we train them not to respond strongly to self-antigens, right? And those antigens are things that are in us when we're developing as young people and not infected. So it's more the steady state, right? Of training our T cells. But the idea here is that there are going to be times when we will be infected and making some additional proteins that are in response to inflammation. So we have to be tolerized against those things as well. And that's kind of the idea of this paper in general, is that this interferon signature in the thymus is really, really important for selecting, and this is kind of like the end result, things like regulatory T cells that are going to allow us or facilitate a lack of response to the inflamed self. And that's kind of the key takeaway from the paper.
Do we know what the signal to produce the interferon is? You said it's not based on microbes at all.
Mm-mm. They just know that it's expressed in AIR-positive TECs. So the AIR-positive TECs are the ones that can also express, of course, the tissue-specific antigens. So yeah, they did all the experiments to show it's sterile, but we don't know what's driving the response.
And AIR stands for autoimmune regulator, this transcriptional regulator. It really is playing a central role in immune tolerance, what you described, and these tissue-specific antigens in the medullary thymic epithelial cells, MTECs, they describe. And so this is essential for central tolerance and preventing autoimmunity. And so what they found in this paper that it was AIR-dependent cross-presentation of antigens, which is really fascinating.
Correct.
Yeah, it's really fascinating. But yeah, I also, am interested in what that signal is. Because, my first thought, for obvious reasons, is that perhaps, there's something going on with, nucleic acid. Maybe there's transcription, say, of endogenous retroviruses or something via AIR. But to be honest, when I first started thinking about this and this idea of type 1 interferon in the thymus, I was like, it's something about nucleic acid. And again, I think about that for rather obvious reasons. But
is the obvious reason because your lab studies it? Is that what you're saying?
Yeah. That'd be why. That's the obvious reason that I often am thinking about DNA sensing.
Yes.
And my first thought was, well, yeah, there's a lot of DNA damage in the developing thymocyte. So that must be it. And yet it's not the thymocyte. It's the medullary thymic epithelial cell. And I think that what that signal could be and how AIR, which we usually think of as sort of a transcriptional regulator of tissue-specific genes, is also regulating this mystery process, is a really cool question.
Yeah. I mean, I think this paper really has kind of brought this idea to life, but really sets up a lot of, I think, important future studies to really understand this. Because one thing that they don't actually talk about really in the discussion of this paper, but I think has been appreciated, is that some people who had severe COVID, like hospitalization level COVID, actually also had anti-interferon antibodies, right? So, how is this related to that? Were these people not tolerized against type 1 interferons? Because that's the idea too, is that some of the products of interferon and interferon signaling, they may be playing an important role again in terms of tolerization.
Yeah. I very, it makes me much more intrigued by some aspects of AIR and what AIR is doing, because I know that there's always been sort of that idea of AIR seems to be a transcriptional regulator, but doesn't quite ever act as a transcription factor in assays.
Yeah.
Or I think, as someone once said to me, it looks like a duck and quacks like a duck, but it's not a duck. And so I almost wonder if by figuring out what AIR is regulating in this context, that might help us with a bigger picture of what is AIR doing overall. And
it's funny that you bring that up. That's how we've described AIR for a very long time, right? So, I guess this does beg, the opportunity to study more in the system.
So I know the AIR deficient mice are out there. And of course, we can think about APESED patients who have a lack of AIR as well. And so now I can imagine doing studies in both of those contexts.
Is there any evidence that other cells produce interferons in the same way, sterile production, like these thymic cells?
Yeah, so they were using reporter mice, and that's how they attributed the expression to the AIR positive techs. But they were really focused on the thymus. They were, and very early, so these were in young mice, right, who were doing undergoing a lot of developmental changes. So, it was just using those reporters that they found expression. But of course, the other cells were responsive to the signature, right? So they were, the antigen presenting cells, for example, so the B cells, the various types of dendritic cells, they were all expressing ISGs. So they were responding to the signal from the type 1 interferons. And it's type 3 too. They were kind of studying both. So,
they say that this constitutive exposure is needed for a diverse T-cell receptor repertoire. So what happens in a type 1, if not knockout mice, type 1 receptor knockout mouse? Does that not happen, or is it?
That's a great question.
Yeah. So one of the experiments that they did towards the end of the paper was they took cells, T cells that developed outside without type 1 interferon signaling. So they were deficient in the ability to respond to type 1 interferons. So they grew up in a type 1 interferon deficient environment. I guess that's what I really need to say. And then they adoptively transferred those T cells into recipient mice and then provided some inflammation. So they used poly-IC there. And what they found was that compared to wild type T cells, those cells were responding more. So the idea was they did not undergo the proper selection, and were probably responding to self. The problem, where you run into the wall with some of these experiments is, we don't know what exactly these T cells are responding to, right? So we can't really probe that very deeply.
Yeah, that's sort of the question of, did they need the interferon when they were growing up, or did they need the interferon when they were making that response?
Right.
They remain interferon receptor deficient in both parts of that experiment.
Right.
So it's really definitely tricky.
Right, to disassociate those two things, correct.
So some work we did years ago with poliovirus suggested that there's quite widespread low-level production of interferons in multiple tissues. So if you take mice which have the poliovirus receptor and you infect them, the only place the virus reproduces is the brain and spinal cord. No other, yet the receptor's everywhere. And if you take IFNAR mice and do the same experiment, IFNAR with poliovirus receptor, the virus reproduces everywhere. So the limitation is interferon. It's apparently there's a low level that is keeping poliovirus down in most tissues.
Yeah, I know that there's some papers from the Belcade lab that I know the best that talk about things like ERV transcription and how that leads to kind of a tonic interferon production. Yeah. I think there are some other, so yeah, that absolutely is sort of a background interferon level. But I think that is partially why this is such an important finding, because that means that when our cells are are outside the thymus, they are constantly responding to antigen in this interferon context. And so they should learn about it in the thymus. And it's so cool to think about, this is how they're actually learning what the interferon signature looks like, that they're going to be seeing in the rest of the body.
What's interesting is that if you take the kidneys from from regular mice with polio receptor, just make cell culture out of them, there's no more interferon and they can be totally infected. So in the tissue, which suggests that the kidney is not the source of interferon, right? Maybe it's circulating, I don't know. But that's why for years people made polio vaccines in monkey kidney cells. No monkey ever has poliovirus in its kidney. Yet if you take the kidney out and put it in culture, boom, completely susceptible to infection because there's no more interferon. So something cool is going on there. It's neat.
Yeah, and the other aspect of this interferon receptor dependent T regulatory cell development that I really hadn't thought much of, I think is interesting. And they call that out too, is there's definitely a lot left to understand.
Yeah, I think it just is really setting up some great future studies. And I'm always kind of thinking about papers to talk to immunology students about that really kind of excite them in a way that we're talking about these big picture things in immunology. And this was a paper that really drew my attention for that reason. So I was happy to share it.
Yeah, just even as you were talking, there are a couple of times where I was like, how am I going to work this into a lecture? Because I want to start thinking about how to work it into a lecture. So I completely agree.
Yeah. And Kim, you're doing thymic work in neonatal pups, mouse pups. And you were describing when we were talking that it's a skill that your graduate student particularly honed over time, but these are very small structures to pull these thymus out from the mice. And so I didn't know if you wanted to talk about this paper in relation to your work in your lab.
Sure. So right now I'm working as part of a PO1 research project with a group of researchers who would probably, if they had to categorize themselves, would say that they are developmental biologists who study the thymus. So they are very interested in thymic epithelial cell populations. Of course, they're interested in T cells and some some selection, post-selection events. But the reason why they brought me on board to help them with some of their experiments is to look at functional consequences of a dynamically changing thymic microenvironment early in life. So they were able to demonstrate through a series of very intense transcriptional analyses of different populations of cells in the thymus over time. So from birth to about 3 weeks of age, that there's lots of changes that are happening in the thymic microenvironment that are impacting the phenotype and potentially the function of lots of cells that are impacting T cell development. So we were working with them to see what happens if you have T cells that only develop in those specific times in the thymus, right? So if you were, for example, to use an animal with a persistently fetal thymus or one that had been thymectomized right at birth, how does that change the T cells that are coming out? And that's the work that we've started to do with that group. So that's kind of the limitation of my thymus development, but I'm learning more. Always learning.
Well, thanks for that, Kim. That was cool.
Yeah.
David, you have a snippet for us as well.
I do have a snippet, and it's a true snippet because the paper I selected is a 2-pager kind of perspective paper, and befittingly, since I'm a parasitologist and not an immunologist, it's on malaria, but it's at least immune adjacent since it's on the vaccines that we have out and potential use in pregnant women. So the vaccines there's 2 of them that are approved. They're being used in the field. They target a molecule called circumsporozoite protein, which is on the surface of the form that's injected by the mosquito. And the thought here is that it's easier to stop 10 to 12 of these forms called sporozoites on their way to the liver than it is to stop 1 billion parasites that are inside of red cells later on. And the vaccines work. They're not great. They're probably in the range of how good the average flu vaccine is. So 35+% or so. And this is measured in terms of ability to avoid severe disease. So it's not measured as infection. But it does work. The response is not all that durable, so you probably will need a booster every year. And I should say that it requires 3 shots 4 weeks apart initially, which I think that would be a tough sell here in the U.S. But imagine in a low resource country where you have to get back out to that village that's 20 kilometers from the main road 3 times, at a year, really.
Yeah. And you're saying following up a year later is a year from the first dose or a year from the last dose? Because 4 weeks apart, really, you're then 3 months down the road.
Yeah.
I don't honestly know.
Okay. Either way, it's significant, but
yeah. So
do we know, are these vaccines specific to Plasmodium falciparum, or do they have any protection against other Plasmodia species?
Very good question. They're specific to falciparum. So they target this protein, and it's actually combined with hepatitis B surface antigen, which can kind of form a particle. It's fused to it, and then it kind of self-assembles. And so the vaccine is good enough that it could potentially save a lot of lives. And so that's why it's being rolled out. This paper really concerns the potential use of it to protect pregnant women. And I'm going to have to digress a bit and explain a little bit about immunity to malaria. So people in endemic areas, they're at greatest risk of death up to the age of about 5. And then for moderate disease for another 7-8 years. And after that, they really don't have much risk of severe disease or moderate disease. They can be still parasitized, and a very good proportion of adults are. But their immune system, and it's primarily antibody-based, the immunity, but it's learned all the different flavors of the parasite. And a lot of this is due to the variant antigen that the parasite puts up on the surface of the red cell. And it's got 60 members of these, and it only expresses one, so it can change disguises. And different isolates, even from the same village, will have a different set of 60 disguises. Where pregnant women come in is that there's one unique member of this family. Kind of ironically, it's somewhat conserved enough that we can identify it in all isolates. And that one is only expressed in pregnant women because it encodes, it's of course expressed up on the surface of the infected red cell, and it encodes a protein that can bind to a receptor that's unique to the placenta. So non-pregnant women, children, men will never see this protein and have no antibody, immune response to it. So a woman that's perfectly immune to malaria, as immune as you're going to get in an endemic area, gets pregnant for the first time, and all of a sudden she's got malaria again. And you have parasites that are sequestering within the placenta. There's mononuclear cell infiltrate, kind of a nasty environment that can damage the placenta and inhibit exchange of nutrients, gases, waste, resulting in low birth weight children, which is not a good situation in a low resource country. So the thought is, can we use this vaccine? The problem is, I mentioned that the the protection by it is not all that durable. So their conclusion is that you'd have to kind of start with women that are of reproductive age and immunize them, and then keep immunizing, with a 3-dose regimen, and then immunize them yearly. It's a bit more complicated, though, because you'd also need to take care of any existing infection, because adults can be infected kind of asymptomatically. So they're controlling the infection, but they could remain infected for potentially years. So you need to drug treat them and then vaccinate. And this would have to go on for their almost their reproductive life. Now, without vaccination, women, once they're in their third pregnancy, have a pretty good level of immunity to this one unique variant antigen. So after that, they are protected from placental malaria. But the problem, I think, and they don't mention this in the paper, but it seems to me that if you're reducing the parasites that get in during pregnancy, you're reducing exposure to this one unique placental specific antigen, and it's going to take that much longer to get natural immunity to it. This is my guess.
Yeah, I was going to ask. So the idea here is to give these women the same traditional vaccine, or would it be a vaccine that also includes this unique antigen?
We don't have a good vaccine against this unique antigen. It's called VAR2-CSA. And there's a whole bunch of other interesting things about VAR2-CSA. It truly is a unique member of this variant antigen family, but we don't have one that elicits good protection directly against VAR2-CSA. So it would just be this same circumsporozoite-focused vaccine.
And is that because the immune response to that antigen is poor? It is not as immunogenic as other proteins, or?
So what's interesting is you can get an antibody response to this in the putative vaccines. It doesn't seem to be as protective as the antibodies that are elicited by a natural infection. So they're just not as good at protecting. And there's complicating factors here. One is the fact that this is a really big protein. It's 300 kilodaltons. We've only recently gotten an idea of what the 3D structure is from cryo-EM. And we've done some mapping of antigenic areas, but I think that's been incomplete.
So maybe there's another part of this very large protein that could be a better candidate, more immunogenic, but that's still in the works.
Yes.
Right. Okay.
Yes.
And I'm just curious, what is as we hear more about federally funded science decreasing. I'm worried about problems like this that are low and middle income countries. And not only that, it's a large population of people, but it's even smaller in terms of a market share if you're trying to convince a company to invest in making this protein for pregnant women to have this vaccine. So, it's just even more concerning because companies this isn't a moneymaker for them, but of course this is a human need. So it's been on my mind lately. So I wanted to bring that up.
Yeah, I think there's a concern now about will diseases that don't happen here continue to be funded?
Yeah.
And I would say that malaria as a whole will probably be okay. And that's because the military has always had an interest in vaccine programs. In fact, the RTSS vaccine, which is the first one that was on the market, and this is the one that targets circumsporozoite protein, that development of that started at Walter Reed, and along with GlaxoSmithKline, and they worked on it for years. And you could probably have an interesting podcast just on all the different adjuvants they tried, because the first formulation was terrible. I think they probably thought, we'll take this part of the malaria protein, we'll bang it together with hepatitis B surface antigen, and we'll have something that's just awesome. But it was a dud. And I think they're on 4th or 5th different adjuvant.
Wow.
The current one is AS01. And everyone's nodding their heads. Okay, everyone knows that one.
But can you explain what that is? Because our listeners might not.
I will turn it over to to an immunologist.
We all nod our heads because they know sort of what it is. It's like
aluminum salts. Yeah.
Yeah.
So I think also the Gates invests big time in malaria. So I'm sure their support will not decrease. They know they know the right thing to do. So that for malaria anyway, you're right. That's probably but there's a lot more.
Right.
Yes. Yeah. Absolutely.
And I think that's an important thing to think about or to educate the public about it. I think some people think that if we stop government-funded research, that industry is going to pick up their share, right? So that they are going to invest in all of our research. But that's not the way that this works, right? And as Stephanie mentioned, they are not going to want to touch something that's not going to be a money-making possibility for them, right? They want the way that it's always been is that the basic research is done in the academic labs and then through partnerships, right, that can become materialized.
COVID is a great example. We had this NIH Moderna partnership, right? Otherwise, maybe it wouldn't have happened. Who knows? It was very expensive to do. So Moderna was a small company. I don't think they could have done it on their own. Yeah. So
I wanted to actually go back to that adjuvant because it's not the aluminum. It's
liposome?
No, it's a mixture of like a saponin, which is some sort of detergent, and then monophospholipid A.
MPL.
MPL.
Stimulates TLR4?
Yeah.
Yes.
But the take home is it's all about the adjuvant.
Yeah. Well, different adjuvants have very different effects. The receptors that recognize them are on different cells. They induce different types of responses. So yeah, looking at which adjuvant works best for a particular vaccine is important.
Just understanding adjuvants in general is so important.
Yeah. And there aren't that many approved for use in human vaccines. So the main one is alum,
especially in the U.S.
Yeah, in Europe, they're a little more tolerant. We're not.
Right.
And I think it's a target if you have an adjuvant in your vaccine, unfortunately.
Right. Well, so sorry to take us down a little side street, but I'm glad we did. But David, to go back to what you were saying, that you have this variant that can infect the placenta, it can cause problems with mom and baby. And so feel free to continue on your path.
Okay. So I think one of and this is getting more into evolutionary biology, but one of the interesting things about this protein is that it's been found in great ape parasites, implying that the common ancestor of the human Plasmodium falciparum, which split off from a common ancestor that now infects great apes had this as well. So we don't actually know that it allows great ape parasites to sequester into a great ape placenta, because that's kind of hard to work with. Not the animal model you want, but it probably that's what it does. And so this has been a strategy by the parasite for quite a while. And when I first got into working on VAR2-CSA, a colleague of mine that was at here at UGA at the time, Julie Moore, and she'd been working on placental malaria for quite a while. And we had a discussion / argument about this. And I said, I really can't figure out why the parasite would keep this protein that isn't useful in most people. It's not useful in kids. It's not useful in males. It's not useful in non-pregnant women. Why expend the energy to keep this gene around? And her response was that in a lot of these countries, women spend a good portion of their life in pregnancy.
Right.
And this is a great niche for the parasite to hide out in. Now, we've since learned that this protein plays kind of a central role in antigenic variation. It's kind of the central node. So when you switch from one of these gene members to another, you briefly turn on VAR2-CSA and then turn it off and go switch on one of the other ones. So it might be the progenitor of everything in the family.
It'd be interesting to know whether the selection pressure for that isolate, that variant, to maintain this protein would change if the generational window changes. So as societies become more educated or there's more resources, women have fewer children. And I'm just wondering, if you reduce the reproductive lifespan of a woman, would you reduce circulation, therefore reducing or changing the pressures on this particular variant? It's interesting. Then you introduce maybe vaccines, and could you reduce it by those two ways?
That's a really interesting thought. I'd never thought of that or heard of it, but that's a really interesting thought.
So, I mean, really goes back to helping people, have healthcare resources, women's education, all the things that we talk about in global health. Yeah.
Yeah.
And the mosquito that transmits malaria, remind me, its geographical distribution is changing over time because of climate change. Does that increase the potential of it moving to where we would be concerned about it more in the U.S. as a potential disease?
We have a cadre of vector biologists at UGA, and that's a really, really interesting question because, as temperatures rise, mosquitoes have a particular range they like.
Right. And
so it's possible that it could be a win-lose or lose-win situation where some areas the mosquitoes can now survive in and transmit disease, while other areas become less favorable to them. We do have mosquitoes in the U.S. Well capable of transmitting malaria, and in the last year or so, we've had several cases. We had a case in Texas, several cases down in Florida, and this was transmission in the country. Doesn't happen every year, but we've got the bugs to do it.
And historically, we had quite a bit of it as well.
Oh, yes.
Yeah, I think that's something that's often forgotten.
Yes, exactly. The CDC is in Atlanta because that was a malarious region.
Well, anything else, David or Kim, that we forgot to ask you?
I would just like to know, is there any, David, Kim told us a little bit about how the paper she chose related to her research. Is there any relationship you wanted to tell us about?
Yeah. So one of the things I'm interested in with VAR2-CSA is the fact that most isolates have one copy. Some isolates have more than one copy of this, which in a way is kind of weird if you think that it's the central node, because now you've got two central nodes. It's also weird in the fact that the first isolate that was found to have two copies of this is one that's been around forever. Was put in plastic in the 80s and has been there ever since. It expresses both of its VAR2 CSA copies at the same time, which for a variant antigen makes no sense. If you've got disguises, you don't put all your disguises on and then go rob a bank. You rob a bank in one disguise, then you get back to the getaway car and put on another disguise. But so I'm kind of doubtful that it is in fact expressed at the same time. At least my hypothesis would be that's something that happened. This in plastic parasite has lost control of this. And so what we're doing is we're gathering all the more recent isolates we can. We're asking them to determine the gene copy number of this. Our original plan was to use CRISPR-Cas9 to put epitope tags in both of the copies. And so then we can follow it. And we can even do some cool things like we could, if we have a population that some are expressing one, some are the other, we could then select, with flow cytometry just for those expressing one and then look for a rate of conversion to the other one. That has proven exceedingly difficult. It turns out this gene is close to the end of a chromosome. And when CRISPR-Cas9 cuts, and this is work out of Artur Scherf's lab, who was doing some other playing around of VAR2 CSA, their explanation for the fact that they rarely got any editing was that the end of the telomere with half the gene just goes floating off. So what we're going to turn to next is single-cell RNA-seq, which we've got plans to do. We almost have the funds to do it. So, but that should tell us in an individual cell, do you see both transcripts of both? The only complication there is that this gene has a small ORF just upstream of it. And that one is involved in translational control. So just finding that they're both transcribed may not prove that they're both translated in the same cell, which takes us back to wanting to keep banging away with the CRISPR-Cas9 to see if we can't epitope tag them.
And they're too similar to generate specific antibodies?
We could probably do that. I've been avoiding it, but maybe I can't avoid it much longer.
Like a nanobody or something like that.
Yeah, CRISPR-Cas9 is just so cool.
Oh yeah, there's no doubt. It's like so easy to do normally as your DNA floats up. Can you do flow cytometry on the parasites if you had an antibody? Oh, that's cool.
Yeah,
that's
and
neat.
you can sort them. Yeah, we have a great flow center here with a lot of fancy equipment. I don't know how to do, but we have an expert down there, Juan Bustamante, who can guide me through it. So.
Yeah, and he also trained here. He was a parasitologist, so he of had been working on that and now runs the flow core. So he uses all his tricks and skills, as David mentioned.
All right, that is Immune 91. You can find the show notes at microbe.tv/immune. If you have any questions or comments, you can send them immune@microbe.tv. And as I said earlier, we'd love to have your financial support, microbe.tv/contribute. Our guest today from the University of Georgia, Kim Klonowski. Thanks so much for joining us.
Well, thanks for having it. It was a lot of fun.
And David Peterson. Thank you, David.
Thanks. I really enjoyed it.
Cindy Leifer's at Cornell University. Cynthia Leifer on Threads and Cindy Leifer on Blue Sky. Thank you, Cindy.
Yeah, thank you. That was fun.
Steph Langle's at Case Western Reserve University. Stephanie Langle. Was that on Twitter or Blue Sky?
It's on Twitter. I have a Blue Sky as well. I just joined the other week. So
same name, Stephanie
same
Langle?
Name. You can find me there too. Yes.
Thank you.
Thank you. This is great.
Brianne Barker's at Drew University. BioProf Barker on Blue Sky. Thank you, Brianne.
Thank you. I learned a lot.
I'm Vincent Racaniello. You can find me at microbe.tv. Music on Immune is by Tatami. Thanks for listening to Immune, the podcast that's infectious. We'll be back next month.